All right. Hello, GLBIO. I am Sania. I am from the University of Missouri. I work with the Digital Biology Lab with Dr. Trupti Joshi. And today I'll be presenting G2PDeep-v2, which is a web-based deep learning framework for phenotype prediction and biomarker discovery using multi-omics data. Before starting, this is the overview of what I'll be going through. So I'll first tell you what and why we're doing it, what was the first version of G2B Deep about, what did we do in the second version, what are the study results from our platform, and the website component of it. So to begin with, GWAS, which is genome-wide association studies, has been around, and it's a powerful tool to study genetic relationship with the phenotype, specifically the SNP markers, which in turn help researchers understand what are the gene markers that are associated for the diversity in phenotypes. Like mentioned in the first presentation, we have a lot of data being generated by the UK Biobank. We have now large amounts of data, and so we can tap into more complicated techniques like deep learning. Turns out we're not the first ones to think about that. There are a lot of fancy packages. A couple of them were mentioned before me. The issue is that even with those packages, you still have to invest some amount of time into setting those up, maybe looking for more than one package to solve your problem. The other day, I was sitting here and somebody was talking about how they couldn't solve their bioconda environment. And I guess we've all been through that. So those kind of problems still exist with those fancy packages, and there is still a lack of web-based services which would provide an interactive web interface to handle that kind of problem. Also, with the increasing size, for example, the SNP datasets is really large. It kind of exceeds the capacity of a normal computer to do those things. And like the last presenter told us, not all of us have access to high-performance computing resources. To solve this problem, we built G2PDeep-v1, and it is as simple as it looks. So a person is able to create a data set. This is only SNPs. So they can create the data set using SNPs. They can build and train their model. This model was based on convolutional neural nets. It was a deep learning model. And they can do prediction and discovery with it. Since then, we've started working on G2PD and it has, as you can see, it has gotten fancier. We've introduced a lot of things in this particular version of G2PD, and I'd like to explain the features of that. So as we can see, we have more omics data available. So the first version was just about SNP data. Now we can integrate gene expression data, miRNA SNPs, DNA methylation, protein expression, and CNV. When it comes to the deep learning side, we have our own model, which is the convolutional neural net-based model. And we also improved upon that from V1 to V2. And we also integrated state-of-the-art machine learning models that you can use to compare your results with. So when it comes to the modeling part, so anybody who's worked on deep learning knows that hyperparameter tuning is crucial to running the model properly, which means that you need to have some knowledge of what the hyperparameters of the model are and how to tune them. And if you're not aware of them, you have to spend a lot of time understanding what these parameters are about. So to solve this problem, we introduced three options where you can run the model with default parameters. But you know what they say, do not trust the model that runs on default parameters. So you can either customize the parameters if you're aware of how it is supposed to be done. Otherwise, you can just select the option of automated hyperparameter tuning, which is based on Bayesian optimization, and that will do the job for you. When it comes to the training strategy, G2PD-v1 was just working with regression. We have incorporated classification tasks as well in G2PD-v2. And for training information, so one would argue that, if you're doing all the job for us, it is like a black box, but that's not true. You can actually look at the model details, how the learning curve is performing on training and validation, and estimate the training time. So you will have open access to all the hyperparameters that are adjusted by the tuning for you. Then for inference, we actually extended the saliency maps to enrichment analysis, which means after you get the output of prediction, you can actually use GSCA to do the functional enrichment within this platform only. This is the backend of the platform that we have, and I just quickly go over this. So this is a web interface that you get to interact with, and you have data sets, experiments, prediction, discovery, and an extensive user guide that will help you through the portal. Then we have the front end interacting with the back end using RESTful APIs, and then we have the core, the task scheduler, so that multiple users can train multiple models and to handle that part of it. So to test out things on the platform, we use the data set from TCGA, the Cancer Genome Atlas. We had data for 6 types of omics as mentioned here. We designed the phenotype, so we had information of how long did the patient survive after the first diagnosis. We capped it at 3 years. If a patient survives 3 or less years, it's non-long-term survival, and if it survives more than 3 years, it's long-term survival. Patients without follow-up information were excluded. We ran this prediction on 23 different types of cancers, and I'm going to share the results of one of the results that we have from one of the studies that we did. So the models that we ran the prediction on were our model, which is the convolutional neural net model. As we can see that we have multiple CNN layers, and at the third stage, the representations are actually concatenated, and then there's a deep neural network, and then we have prediction and evaluation. The machine learning models that we compare a deep learning model with are decision tree, random forest, SVM, logistic, and they're like commonly used in a lot of all the domains that we can see here. So here are the results, and we ran the predictions on 5-fold cross-validation and averaged out the area under the curve ROC score here, where we did it for skin cutaneous melanoma. And here we can see, so if we look at row-wise, the highest score is the one that's in bold and underlined. And you can see that in most of the cases, multi-CNN is doing a great job of predicting. We have compared results from 1 omic to up to 3 omic, because the model is capable of running from single omic to up to 3 omics of data. And the idea of this slide is not to compare the performance of models and say that our model is doing best, but it's what a user can do with the platform. They can compare omic-wise results, and they can also model-wise results and see if deep learning is actually the way to go for their kinds of data. After getting the results for this, we run saliency maps, and by saliency map algorithm, we get a list of ranked genes that's best at differentiating between the two classes, long-term survival and non-long-term survival. We take the top 100 genes from our results and intersect them with the oncogenes, where we can see that 14 of them for skin cutaneous melanoma actually intersect. We're also able to run GSCA with KEGG and Reactome, and we get some of the pathways that we can actually see are supported by literature. Now, moving on to the website component of G2PDeep-v2. This is the homepage, and we can see that all the introductions that we went through in these slides are actually there. The web address is g2pdeep.org for your information, so that you can visit and check it out. So a person has a very different space. Each user has a very different space, which means that their models, their data sets are private. The features are mentioned on the left-hand side, your right, my left. And then you can see that we have a workshop tab. So we conducted an ECCB workshop in 2023, a very detailed account of how this works, what led to this. The technicalities behind the deep learning models is all there. So you can all check out the video. We have a very extensive tutorial section which you can use to see how to do things, how to upload data. So in the dataset section, you have the option of actually adding plant, crops, human. A lot of organisms data can be uploaded, and you can try different phenotypes data as well. We have a public dataset section. We have uploaded a couple of public datasets, and if you do not have anything and you would just like to play around with the data, you can use that. Otherwise, you can create your own dataset, and that would be initially private, but you do have the option of making them public if you like to do so. Once you go to the model creating part, you have the button where you click to create the model. The model will, like the dataset, will also be private, and you can make it public if you'd like to. Once you click on the button, you select the number of omics that you would like to perform the prediction on. What's your task like? Is it classification? Is it regression? And then you would like to choose tuning techniques that you want to do. So here is what we talked about, the automated hyperparameter tuning. Once you've done that, you click OK, and then you will have the model. Once you do that, you can train your model with the data set that you uploaded, and these are what the results can look like in one platform. We can see how the model performed, the learning curves on training and validation, how did the losses look like, what was the distribution of our initial data, and how did the distribution of the predicted labels look like. And we can also do gene set enrichment analysis here in the same platform. So since we launched G2PDeep, we have around 600 total projects. We have 60 models and around 200 data sets up to date. So this is the current state of affairs with G2PDeep-v2. We've already started working on G2PDeep-v3, and we plan to extend the model to single-cell multi-omics data. We also want to handle the class imbalance problem because in a lot of places, the data set, for example, the positive disease class is really low in number. And so the model results tend to be biased towards the majority class, and we'd like to handle that in our platform. We want to use large language models to generate multi-omics embeddings to make the prediction and see how that goes for us. We want to extend the model. We're already doing that for crops. We want to do it for viruses and other organisms as well. And we would want to further improve the explainability of the model. Last but not the least, I'd like to thank my team, Dr. Joshi, who's here, and Dr. Shwai. Without them, it wouldn't be possible. So thank you so much. And I'd like to thank NSF and GLBIO for awarding the Travel Fellowship. Thank you so much, and I'd like to take any questions if you have. Yes.
So for the saliency analysis, is that just for the CNN?
Yes.
Do you have any other, like, I'm curious if you're planning to do interpretability for other kinds of models that you make available. Like, would you do a comparison of those between different models, or like, are you going to qualify that based on the accuracy of the models?
So it's already there. The interpretability in other models is already there. We do it with the coefficients of the algorithm or the model. The chain model coefficients are used for the ranking of the genes. And that's how we do it for the other machine learning models that we have. And that's how we get the gene list. And that's how we do the GSCA part. Yeah. So I hope that answers the question. Yeah. Okay.